The standard vision of science is of a very arcane technical craft, impossible for many people to understand. It's a view of the world that is uninspiring and, in many people's idea, lacking all creativity. Yet my experience has been just the opposite. I found science to be fascinating and creative and hope to convey some of that to you. Back to the arcane, a jellyfish. Initially, there was a scientist who studied this particular jellyfish for about 20 years. He and his students collected about 1 million of these over that time period simply to understand why they glow green. And what he found was, this was the reason. They have a protein called green fluorescent protein, or GFP for short, that glows green when exposed to blue light. And the arrow points to the part of this particular protein that actually is involved in the green glow. Other researchers had used this green fluorescent protein to express it in mice and other creatures and learn what they do. And they also used it to paint cells to try to better understand their function. And this particular picture is of something called a brainbow, a rainbow-colored series of cells from the brain that are used to identify the function. This particular slide is an illustration of the use of this protein to understand cancer. The picture on the right shows this green fluorescent protein and the detail in a part of the cancer cell that you didn't see from the traditional technique on the left. Over time, many different colors have been produced other than just the green, and this image shows bacteria containing the different proteins, green and others, that were put on a petri dish and then exposed to blue light, a scene from San Diego. This next example shows again another use of this green fluorescent protein and other variants. These are fish called glowfish, and they originally developed to study pollution. They're now sold as aquarium fish under that trade name glowfish. Coming back to this theme of creativity in science, as with the famous story of Isaac Newton observing an apple fall and describing gravity as a result, creativity has been a key element in science. It wouldn't have been easy to tie together this jellyfish protein and medical advances, but that creativity ultimately resulted in a Nobel Prize for several scientists. Another example of creativity is using nuclear testing as a means to better understand how the brain and actually other parts of the body function. And what scientists realized was that the tests that had been done, the nuclear tests that had been done above ground in the 1950s and 1960s, had actually created a kind of signature or stamp that could be used for better understanding the brain. And in particular, one question regarding the brain was, do we make new brain cells during our life? The creative scientists involved in this project realized that the radioactivity from these nuclear tests essentially created a kind of date stamp, so that cells that were born back in the 1930s had no radioactivity. Cells that were born later, for example in this graph, in the 1970s, would show quite a bit of radioactivity from those nuclear tests. And even though they had come up with this creative idea about using fallout from the radioactive tests in order to try to understand something about brain function, they had a lot of work to do to try to translate that into experiments that actually produced a result. The amount of radioactivity from the fallout, thankfully, was 50 to 100 times less than normally used in the instruments that were available. But they applied their creativity again and were able to get an answer that ultimately, in terms of results, that we generate only a little less as we age, about 700 new brain cells per day. So for those of you who think you're getting older and the brain's not working as well, not necessarily the case. You're making some new brain cells, probably as we sit here right now. Those of you who are Monty Python fans, now for something completely different. Thermus aquaticus is a bacterium that grows in the hot springs in Yellowstone National Park. It thrives at about 160 degrees Fahrenheit, and scientists who studied this particular microbe realized that its ability to thrive at high temperatures made it capable of performing some unusual feats. What's shown in the left part of this slide is a molecular copy machine technique that requires a high temperature enzyme, shown in the right, that's used for a number of different purposes. For example, identifying human remains, identifying criminals from their unique genetic fingerprint, testing cancer cells to determine treatment options, testing donated blood for HIV infection, determining whether a person has a TB infection, sequencing the Neanderthal genome, and a number of other uses. The realization that critters that live at very high temperatures had valuable enzymes and other properties led to extend this search. And one example was looking at hydrothermal vents under the ocean. The creature in the upper left-hand side of the screen that you just saw was a microbe that grows best at boiling temperature in water. So back to Isaac Newton. I hope I've gotten you thinking that science is indeed a creative enterprise, whether it involves watching an apple fall, studying jellyfish, or collecting samples from a deep ocean hydrothermal vent. I also hope that it's given you some food for thought if you've had, somewhere along the line, a bad experience with science. My experience has been that science is fascinating and creative, and I hope that it will inspire some of you, this presentation, to give science a second chance and look at the creativity behind some of the discoveries that have been made.